All right, good to go. So we're gonna first do, I don't know why I'm standing, I'm just anxious right now. First gonna do lecture 1, which is talking about the skeletal muscle. And so there was 12.1 from the book and 12.3 if you wanted to read the book, but honestly the book's exactly the same as her lectures. So there are 3 types of muscles. There's skeletal, cardiac, and smooth, right? We're mainly going to focus on, in this part of the lecture, on skeletal and smooth. Smooth is going to come at the very end. We're going to focus mainly on skeletal. Now, skeletal muscles are striated and multinucleated cells, right? So there's actually a really good picture right here that shows what I mean. So for example, right here when she shows your, I think that's a quadricep, right? No. Anyways, it's close to the bicep. It shows a striation and it's multinucleated, so you can see there's multiple nucleuses intermingled with one another, and they can communicate by what we do is called a neuron. There's 2. I'm doing this really bad. I'm sorry. I'm really anxious. I should probably just follow one. All right. So there's something called the origin and insertion, right? So the origin, you can think of it as the point of where the muscle is actually tied to the bone, right? That's the original or origin of the spot. Right, and I wrote right here in my notes, where did I put it? Oh, it's the closest to the trunk. So you could think of this, and I wrote it as an example as my brain, you could think of this as the trunk, which is your bone, as like the main base, while the origin is like the stem of like a tree. It's popping up, right? And the branches would be your insertions, the more distal part. So for example, if you had a bicep, right, your insertions, right, or your trunk's the bone, the insertion's the first part where the tendon is, and like some muscles attached, and then at the very top of the bicep, right, that's your distal or your insertion point. And then it follows suit on the other side, right? You have something called a flexor and an extendor. Now, easy way to remember this is flexor brings your bones together, and extendor, it moves it away. But you could also know it as this. You want your X to go away, and your extensor moves your bones away. Cass is the only one here. I feel so bad right now. She's enduring these puns. But if you remember it that way, you could know, like, okay, I know extensor means to go away because of the X thing, so I know flexor has to bring them back, right? So here's an example of that. When you're flexing, you're bringing your muscles in, and then contracts your biceps. And then when you, stretch, you have your tricep contract and your bicep relax. And that pushes the bone away. Now you have to remember, every muscle has the opposite form of it. So for example, for this, it has antagonistic form of it. So for example, for this one right here, if you only had your bicep and you flexed it, your arm would stay like that forever. So you needed a secondary antagonistic muscle to move it back out, which is your extendor, aka your tricep in this case. Cool? Cool. Every muscle has that. So, let me just, flip it to script. Each muscle has something called muscle fibers, right? The muscle fibers is the smallest unit of your muscle. Cool? And they're long, cylindrical, and they're made of they're many fused nuclei that are all together. So think of them as just an easy way to understand of a muscle fiber. Just think of it as like a small cylinder that you're just stacking all above each other, right? And in the cylinder, since they're all touching together, you can think of them as technically one big superstructure. Cool? You also have something called satellite cells. Basically, these are just growth cones in your muscles. So like, you're going to work out, right? You're doing, for example, let's say push-ups, right? So you're working your biceps as well. So when you do that, you're actually tearing some of those muscle fibers. So what happens is these satellite cells, aka they're pluripotent stem cells, specifically for your muscles. And they look for those ripped up parts and certain parts, and they just produce more muscle and more growth, which allows your muscles to grow bigger, larger. But it also occurs when you're going through puberty or anything that has drastic change to your body structure. So it works for if you're doing like some weight training, like an athlete, or you're just growing up. Cool? You lose actually some satellite cells when you get older. The older you get, the less of them are actually in your body. So like she mentioned, I think at like the age of 30, that's basically your drop-off point. So trying to work after age of 30, you have much more or less satellite cells than you did at like say 25 or 28. Cool? So this is the basic structure of a muscle cell, and I wrote how like a good acronym for you to get. So I got EPIMIS, right? And going EPIMIS, I know it's like I'm gonna go from back to forward, but it's easier to remember this way. So first starts off as your sarcomere right here, right? In the muscle fiber, there's a sarcomere that has everything in it. So it starts off with sarcomere. That's the S part of EPIMIS. Then it goes to your muscle fiber. So it's sarcomere, muscle fiber. Then it goes to your endomysium. So you have your muscle fiber, and then the endomysium is just like, think of it as your like you have your U and you put an undershirt, right? That's your undershirt. It just covers it up. Now when you have a bunch of endomysiums together, you actually wrap it up in something called perimysium, right? That's like if you put a shirt on, right? And then finally you have something called epimysium, right? And that's like the overall bundle that holds them all together. So that's like you just put on a big sweater or a big jacket and you want to zip it up, hold everything together, right? So I wrote it, you could write it as the reverse, which is S-epim, but I put epimis because it's actually easier and it rolls off the tongue, and you can just remember the like reverse or forward way. So you can be like, oh, it's epimysium first, then perimysium, then endomysium, then muscle fiber, and then sarcomere. Cool? And then she can ask you, oh, what is this? You can just remember that acronym and be like, oh, I remember epimysium, epimis. So all of this actually allows you, so all those categories together actually allows your muscle to actually contract and produce a force. So that's like why that's important, because each one, if they both don't work in tangent together, you won't have any force. Cool? So here's a beautiful chart you can do at your own time. Really, you don't have to look at it. I mean, I'll break it down. So here's some terminology that's similar it's exactly the same. They just have it specialized for muscle cells, or skeletal muscle cells, I should say. So your muscle cell, for example, it's called a muscle fiber instead. Your cell membrane is your sarcolemma. Your cytoplasm is your sarcoplasm. And your modified endoplasmic reticulum is your sarcoplasmic reticulum, or SR, which will be later referred to as. Cool? And I'm pretty sure the professor as well, during class, she just says SR now. She doesn't actually say sarcoplasmic reticulum each time. So you at least know what that means, right? Cool. All right, so here's the basic drawing of the skeletal muscle. Now it's very simple. You have a neuron, and in this neuron, right, you have your alpha, or your upper motor neuron, connected to your central nervous system, and then you have your lower motor neuron, which is connected to the upper motor neuron. So basically what happens is my brain sends a signal, and then it travels down to the upper motor neuron, which then sends a signal to my lower motor neuron, which attaches to the muscle fibers, which creates something called an end plate potential, right? Or a muscle end plate. In that point, your lower motor neuron actually releases ACH, acetylcholine, into the muscle, and that triggers it binds to nicotinic receptors onto it, which triggers an action potential to occur. This action potential actually travels down something called T-tubules or transverse tubules. So be wary, she could actually ask you, oh, what are transverse tubules? Don't trip out, it's just T-tubules, right? And that'll produce an effect, right? So here's just an overview of, here's the sarcoplasmic reticulum, you got T-tubules that are all interconnected to one another, that's a key point, right? And then you have your beautiful myofibrils. Cool? So here's a key thing that I want you to remember about specifically for this part, right? You have something called thin and thick filaments, each in the myofibrils. Your thin filaments are always actin. No matter what, they're always actin. Now you have something called G-actin molecules, and if you have 7 of them together, they become an actin, like an actual actin thread. Cool? And you also have something called troponin and nebulin attached to it. Cool? And also tropomyosin, which is actually going to be a very big deal later on. So now you have also on the thick filaments, you have something called myosin, right? On myosin, you have first the myosin tail, then the hinge region. So the myosin tail is actually the thickest part, The hinge region is just a small intertwined two-part. And then you have the myosin head. And on the myosin head, there's actual regulatory sites. We don't have to worry about them. It's good to know, like, you got some regulatory sites on it, right? Actin is different, though. Actin, you do have to know the sites on it. So there's troponin C, I, T, and there's also tropomyosin and actin. Now, let me just turn the page here. Here we go. Troponin C binds to calcium. So that's the main part, because calcium actually helps with the process. In its entirety, calcium is the main thing. If you don't have any calcium concentrations, you're fucked, right? And it causes a conformational change. So what happens is, I have calcium and it binds to here. Cool, causes a conformation change and it moves, allowing I to pop out, right? Which allows troponin I to have a conformation change as well, and move troponin T. So basically, all that happens is it's like the wave, you know, like the hand motion, the wave. What happens is someone taps you, aka calcium, taps right here, moves I, which moves T, right? All that comes together and it moves tropomyosin. Where is it? Tropomyosin is this yellow wire. Moves tropomyosin, which actually opens up or frees or like shows the actin is there. Now you need this to happen because if no calcium shows or doesn't do any of that conformation change in troponin and tropomyosin doesn't move, no matter what, you won't have any force at all produced, or myosin will never bind to actually actin. So that's a key thing. So if she says in an exam, something happened, blah, 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 and no more calcium is produced, which there's like another thing we'll come to later, but if no more calcium is produced, it won't cause a conformational change in troponin C, which won't cause change in I or T. So she could ask you a problem like, hey Cass, if troponin I is broken, but I still place calcium into the solution with thin filaments, what's gonna happen? All you're gonna say is, oh, it's still gonna change conformation for troponin C, but since troponin I doesn't work, troponin T won't work, and then tropomyosin won't be removed, right? So it's like she could ask you something dumb like that. Cool. Then after it like frees itself, tropomyosin's free, actin's open. What happens is myosin will actually, let me see if the next picture shows it. It does not. Myosin, one of the heads will actually bind to the globular actin molecule. So these, I'll just point from here. These little reddish balls, or mainly, it's actually going to be like, see these little caps on the red ball? These yellow caps? That's actually where the actin will bind. Right? That's where the myosin heads will bind. Sit down, because I feel like an asshole. Cool. That will make something called a cross bridge. Now, cross bridge is important because depending on how many cross bridges you have, will actually explain how much force you have. So, for example, if I had, let's say I have a thick filament that has 10 myosins on it, and I have an actin that also has 10 actin G molecules on it. Cool. But when I first activated it, or when I first sent a summation of an action potential, I only have 2 binded to 2. It's actually 6 to 1, but anyways. Like the actual ratio is 6 actin to 1 myosin, by the way. Just like for the skeletal muscle, 6 to 1, but for simplicity's sake in the explanation of this, let's just say it's a 1-to-1 ratio. Cool? So I have 1 myosin binding to 1 actin, and I have 10 in total, but I said only 2. You have actually a very weak cross bridge, which will produce a very weak force. You have like, it's like if I told you hold one finger to one finger and then pull, right? You can easily pull them apart. It's like not that much force. But then if I said, all right, pull all 10 with all 10, right? And you put your fingers together, in this case your 4 fingers, and you pull, it takes more effort and more energy to pull them apart, but it gives you a stronger force. That's like the overall underlying message. Cool? What happens is the myosin will always, and I'm pretty sure I can actually show you right here, perfect. What happens is the myosin will pull this actin right here to the M line. It'll always pull it to the M line, right? So it doesn't matter, well, it kind of does matter, but this side right here will pull that actin towards it, and this side right here will pull this actin towards it. Cool? And don't be confused because like the zigzagginess actually, in other videos, she didn't show us in class, but in other videos, basically it just it just gets flattened out when it gets pulled both ways. It's not like the zigzagginess just goes like, oh, I'm still zigzagged, so I can't. It'll just flatten out. So if like you're trying to picture it in your head, it'll just be like a straightish line. Cool? That's just if you didn't know. You will actually have to know all of these zones for the exam. She mentioned it like twice in her video, so I made sure to write that down, that you actually have to know the zones. So it's very easy to remember. From one Z-disc to another Z-disc, that's your sarcomere, aka a what? What did sarcomere stand for? You can look at your notes. I'm not gonna like. One more page. Did I not tell you? How rude of me. Did it not show? I thought it was right here. Oh, the circle on. Oh, I'm so sorry. I didn't. That's on me. I didn't teach you what sarcomere is. It's just basically an entirety between two actin thin filaments. That's what a sarcomere is, right? So you have, it's like from point A actin to point B actin. That's just a sarcomere. I'm so sorry. I thought I taught you that. I just panicked a bit, I guess. Cool? And in between that sarcomere, you have something called your H zone. Now, the H zone is basically just think of it as like the thick filament structure without the actin or thin filaments moving in. Cool? That's your H zone. It's a clear band with, A band, with an A band in it. So the A band encompasses the H band. Basically, the A band is the entirety of the myosin, or the thick filament, right? And then you have your M line, which is just the middle structure of the thick filament. So think of it as like a ring, right? From the end to end of the ring, you have your A band. From just the space, think of it as like the little, like if you had a ring and you flipped it and you're looking inside the ring now, that little gap in between the two parts of the metal, that's gonna be your H zone, right? Your M is just gonna be the spine of the ring. Cool? Your I band, now that's just the spacing in between both A's. So it's very simple to remember. You can use the acronym IRO, but I'm not sure you're gonna like it. But it's basically ZHIAM, which is Zahem. It's not that good, but it's easy to remember because you can think of, okay, a Z disc, and if you remember this picture, a Z disc is just from one actin thin to one actin thin structure. And H is just the gap in between both of the, my brain died for a second. It's just the gap in between the thick and the thin filament. You have to know the I, which is just the gap in between the thick, two thicks together. And then the A is just your entirety of your thick filament, and then your M line is just the spine. You can come up with a better acronym if you like. So here's a better description of the sarcomere, in like a 3D sense, right? So this is what I meant by you have to actually know the bands, because she can actually ask you specific questions about each band, which one each one represents, that sort of thing. Cool? This is a 3D rendering you could look at. It's exactly like this, but it's just 3D version. I won't bother you with that. But basically, if I could zoom in, which I couldn't, what happens is, this is what I was telling you about earlier about the cross bridges, is that this is a thin actin filament right here, right? And what happens is calcium, which will be released from your sarcoplasmic reticulum, right, which we'll go through. Anyways, the calcium will go here and bind to troponin C, which will cause a conformation change in I and T of troponin, causing tropomyosin to move on the thin filament. They're all valves on the thin filament. The thick filament, all you have to know is that it has a hinge region and the heads. Cool? So she can ask you a weird question, right, and be like, troponin on the thick filament, and you'd be like, whoa, troponin's not on the thick filament. So automatically you'll be like, oh, it's E, none of the above, because this can never occur. But anyways, all that will move, and then these actin, right, will bind to the G molecule, this myosin heads will bind to the G molecule. Then that'll pull it towards the M line. Cool? When it's pulling it towards the M line, this, just for like simplicity's sake, it'll just straighten out. So you don't have to worry about like the zigzag thing if you were ever confused about that. I'm just saying it because I was. Anyways, it'll just pull it towards the M line. The more bindings you have, aka the more cross bridges you have, the more force you have. Cool? That's another picture of it. So you have these things called titin and nebulin, right? Titin is your elastic protein. It's the one that stretches. So it's right here on this little guy. Think of it as just like a spring, like you compress and stretch a spring. It's got a lot of potential energy when it's in its relaxed mode, and it just releases it when it's got to be stretched. Cool? It's like a spring. While nebulin is your inelastic, and it aligns the actin filaments. So you have a bunch of actin, right? But not all the actin, if it's not aligned properly. Think of it as like when you were little in kindergarten or something, and you just had to line up. It was like a zigzag. Everyone was like standing next to their friends. They were lined up, so to speak, but they weren't properly lined up. So the teacher would actually, which is like a nebulin, the teacher would actually come and be like, all right, everybody straight line. She'd like straighten out the line. Cool? The titin would be like if she told you guys, hold hands. So like no one gets lost, right? If you're on a field trip, so you'd hold like your friend's hand or the person next to you in the thing's hand, and that you guys would stretch depending on how fast someone was walking or how slow you guys were walking. So you can think of it as like that. So titin would be your elastic boy, you guys are the hand holders, while nebulin is like the teacher telling you guys line up, and those guys were aligned actin. Cool? And it helps align the actin with the myosin as well. It's like a little guiding hand. So here's a good picture of it. Nebulin's right here, and it helps align the actin, and then it binds to the myosin. Coolio? And this is just showing you the relaxed and contracted state of the titin. So for example, here's relaxed. See how it's like all nice and spread out? Like a spring, it's like uncompressed. And then you contract the muscle, it gets contracted as well. And supposedly builds up potential energy. She didn't go too much in depth on that. So this is what I was telling you about when I was telling you about the cross bridges. We'll take a little break, don't worry, as soon as we're done with this. Let me see when it stops. Okay, after this part, we'll take a break because you could like take a breather and whatnot. But anyways, muscles, whenever they contract, they create a force. And depending on how much myosin binds to the actin, will create either a greater or less force. Now you have something called your stretch point, which is the point where you have the least amount of force. So it's like if I had a really tall box. I told you, hey Cass, push this box. And you're just like, okay. And you just like you're barely standing, your arms all limp, and you're not gonna produce that much force, so the box is not gonna move. Now this is what she keeps pointing out in class, but she doesn't call it by this name. There's something called your optimum point, right? And your optimum point, or your optimum overlap, right? That's when you have the most overlap of actin and myosin, like the max point. So your stick point or your stretch point, that's when you barely have anything stuck, right? So you're just like, it's like you just put your hand like that. But your optimum overlap, that's when you have the most myosin heads binded to actin G molecule. And that's like when you're like, all right, I gotta push this box. Cool? So we'll take a little breather.